Hi, everybody, and welcome to the Engineering Tomorrow Drug Delivery Systems Lab. We're going to talk a little bit about how engineers are involved in the pharmaceutical industry. And they're actually involved in a lot of different ways, and we'll talk a little bit about that. But what we're going to primarily discuss is how to design a system for delivering medicines in a very engineering way so that they are taken up by the body in an appropriate way for the patient's health. So let's get started. As you may know, Engineering Tomorrow breakouts and labs have all been developed by engineers. And I worked on this lab together with Maria Krug, who is a recent chemical engineering graduate from the University of Notre Dame, and is currently working at AbbVie doing biologics. But just to give you a little bit of background of me, I am also a chemical engineer. And I spent 17 years with GE, General Electric. And one of the jobs I had at GE was actually in the pharmaceutical business, in GE's pharma business, which made injectables, which were used whenever you're going to have a scan. So if you've ever had an MRI or a CT or an NMR type of scan, quite often a patient will get an injected material. And that allows the imaging system to take better pictures, basically to see more of what's going on inside your body. So we used to make those. And again, you might notice here, both of us were chemical engineers. And chemical engineering is actually an area of engineering that is very well suited to the healthcare industry, at least the pharmaceutical industry, not only in developing drug systems like we're going to talk about, but even more importantly, in manufacturing the pharmaceuticals, manufacturing the drugs, because they typically are chemical manufacturing and packaging in their areas that chemical engineers do a lot of work in. So let's talk about what the lab is about, and we'll start with the conceptual basics here. So pharmaceutical drugs are administered, or they're meant to be administered in a way that has the biggest impact on helping a patient get better. So here are four ways that drugs can be administered. One is very common, very typical, you've probably seen before, and that's just oral administration. So that's basically just taking a pill. So maybe a Tylenol or an Advil or any kind of pill that you have to take. And that basically obviously goes into your digestive tract, goes into your stomach, and then gets absorbed into your body. Another way that it is done is through inhalation. So you may have seen inhalers. That's quite often used for asthma. And essentially then you're injecting the drug or the medicine directly into your lungs. And then it can get taken up much more quickly through your blood vessels in your lungs. Another way is quite often if you're in a hospital and you're on some medication, they'll have an intravenous line in one of your veins. And in that way, they can inject drugs or medicines directly into your bloodstream very, very quickly. And then the fourth way that we show you here it's called subcutaneous injection, but it's basically an injection, a shot. So we've recently gone through COVID, and the COVID vaccines are essentially injected this way, where you put a needle into your skin, through your skin, and you inject the medicine directly into your below your skin tissue. So there's obviously pros and cons of each way. Some of these are easier than others. Some of them can be more dangerous because there's a risk of infections. But they're all very valid ways of applying or administering medicines to help people. So let's talk about how a drug delivery system works. Drug delivery systems are basically the thinking about what's the best way to get the drug to the right place at the right time. And it's really important, obviously, because you want to have a drug that is effective in your recovery or in your treatment of your symptoms. And it's important for it to be safe and to act the way you want it to act. So let's talk about some drug delivery systems. Now, this is actually a video that talks about a very specific drug delivery system. It's research that's being done at a university in Israel. And I know it's kind of a cartoon, but it's a good description of how engineering and medicine can come together to figure out effective engineered methods for treating diseases and for delivering drugs where they need to be delivered.
One of the major problems in injecting medication such as chemotherapy is that the medication affects the entire body and not only the sick cells, and side effects can be very severe. Innovative research is currently developing a cunning drug delivery system. Ben-Gurion University scientists, led by Professor Yossi Kost, are developing a system to sneak medication like chemo or genes past the body's defense mechanisms and straight to their destination, even a cell organelle like the nucleus inside a cancer cell. The scientists are working to create nanoparticles made of a special polymer and a medication that can distinguish between healthy cells and cancerous cells. The nanoparticles are small enough to pass through tiny holes existing only in blood vessels surrounding cancerous growths. When the nanoparticles identify a sick cell by its external receptors, for example, they attach to the cell. But not all of the particles are able to penetrate the cell membrane. Here the researchers use ultrasound in an extraordinary way. They focus low-frequency ultrasound waves on the target spot to make a tiny and temporary opening that enables the particles to penetrate. Next, the particles need to get to the nucleus in order to release the medication. The smart polymer knows how to identify unique intracellular proteins that travel into the nucleus, and it hitches a ride on them. Once the particles reach their target, they need help releasing the medication from the polymer. The scientists again turn to ultrasound for help. The ultrasound waves are aimed at the target cells and the medication is released, while the rest of the body's cells are safe from side effects. The research combines the use of ultrasound, nanoparticles that identify sick cells, and smart guidance to the target point, all in an effort to provide effective healing without harmful side effects.
So what you've seen in that video is it can be pretty complex. Obviously, you want to get the drug delivered to the right place at the right time, and you saw that there are many different ways of affecting that delivery. They were using ultrasound to actually do some very interesting things, not just for imaging, but for actually causing drugs to be released at certain points in time. And it's also important to understand the biology of cells in order for this to work. Now, understanding how medicines or really any component can diffuse through other materials is really a critical thing. And diffusion is quite a key fundamental process that you learn a lot about in chemical engineering, because it's also something that's fundamental to a lot of other chemical engineering processes or chemical processes, which we'll talk about in just a minute. But essentially, for the drug transport, first of all, if we take you step by step, first of all, when you swallow a pill, and as I said, it makes your way to the stomach. It then breaks down, and the drug substance is then absorbed in the stomach, and it's absorbed by blood cells via diffusion and through the membranes of the cells. So here's a representation of your blood, which includes a lot of different things. There's plasma, white blood cells, red blood cells. And if you zoom in on the red blood cells, you can see what we have here, what looks like the blood cell membrane, which is basically what's enveloping your cell, and you want the chemicals, the medicines, to actually go through that cell membrane. So this represents over time how originally there's a lot of concentration of material on the outside of your cell membrane early on, but then over time it slowly diffuses through the membrane until there's an equilibrium. And this is one of the fundamentals of diffusion, is that diffusion is actually the movement of materials or the movement of chemicals, and they move from a high concentration area to a low concentration area. That is what diffusion is. Diffusion, now we get into some real engineering here. The diffusion, as I said, is driven by concentration, so from high to low concentration. And the way engineers talk about diffusion and measure it and model it is through something called Fick's law. And essentially what Fick's law is shown over here on the left. The diffusion flux, or J, is going to be a function of the concentration gradient of the material. So the concentration is this phi, and the change in concentration with distance, or the gradient over distance, is the driving force. And then there's going to be a diffusion coefficient, which is the big D here. So essentially what diffusion is, if you kind of do a thought experiment here, let's say you have a jar of water. If you insert or you inject some dye molecules into it, originally it's going to be very concentrated in the middle of the water. And then over time, those the dye molecules are going to basically diffuse until eventually the whole beaker here has one constant color, because it's been diffused evenly. And now there's no concentration gradient driving the movement throughout the water beaker. So it's basically going to be now at steady state or equilibrium. So again, with diffusion, it's the concentration gradient that's critical. So I mentioned that this is actually very important in chemical engineering, because this is also the fundamentals of how many water treatment processes work, with driving the concentration from a high concentration to a low concentration. But in water treatment, it's actually, we're trying to do the opposite. We're applying extra pressure to reverse that direction of diffusion. Diffusion is also key in the scrubbing of gas, which is basically the purification of gas, which you can see in the center picture here. And then, as we talked about in biological processes, diffusion is quite important, where you can have the diffusion of oxygen in your lungs, which is how your body gets its oxygen. So diffusion is very fundamental process, and chemical engineers work on this and utilize the fundamentals of diffusion in designing reactors and all kinds of processes where we're dealing with materials. So let's talk about a little bit closer to what you're going to be doing in your exercise today. The way drugs are designed is obviously they have to be packageable, that they have to have a package so that they can be handled. Now we're talking about not injectable, but rather pills that you would take. And they're typically coated with something called enteric coatings, which are essentially coatings that allow them to be handled, but that will encapsulate the key pharmaceutical element, or the element that we're trying to have in your body that'll help with any disease or any symptoms you have. So these enteric coatings have to be designed very carefully. And quite often they're actually made out of polymers. And part of the reason polymers are good is because they can dissolve. You can make them in ways that, depending on the polymer, that are safe for the body. And they are basically good packaging type of materials. So you can see an example of what we call an immediate release pill or immediate release bead, where you may have the core, you'll have a drug layer, and then you'll have an overcoating. So when this goes into your stomach, the overcoating will dissolve, and then the drug will be released in one kind of one process. Now, another way of maybe delaying the release or the application of the drug is to have a delayed release speed, where you may have multiple layers of materials. So in this case, you have an overcoating. You may have a slow release delay polymer or material. So that gives time for the pill to get to your stomach or maybe into your further into your body. And then by delaying the dissolution of that, then you can actually release the drug a little bit later in the digestive process. And that's partly what you're going to be designing. You may imagine that you can have a multi-release process now, and that's what a dual delayed release drug delivery system is. Essentially, it's a process where you want to have a certain amount of the drug released early on, and then you want to have the pill still be dissolving slowly, so that several hours later you'll have another dose of the same drug, so that you can take one pill, for the day instead of having to take the pill multiple times. So this shows a little bit about what that looks like. If you have an immediate release, where you just have a single release in your pill, you may have a peak concentration of materials in your body, and then it decays, and then down here you'll have to take another pill. But with a delayed release, you get a single peak for your first release, and then the pill is still working. And then once you break through to the next level, you get a second release. And that's the dual delayed release, which is basically an engineered drug delivery system. Now, here's an example of what that looks like. This is actually a pill or a medicine that's used for acid reflux. And essentially, it is a dual delay release packaging, so that it allows the pill, the first layer of the pill, to release higher up in your digestive system, and then the second part of the pill to be released further down your digestive system in your large or small intestines. So let's get to what you're going to be working on. You're going to be building a dual drug release system. And as you know, with engineering, kind of the fundamentals of how engineers work is that you have a design, you have a model for how things should work, you make your initial design, and then you test it. And if it doesn't work, you learn from what didn't work so that you can get a better design and a better trial the next time around. So hopefully you'll have enough materials so that you can do multiple tries on this. What we've given you here are some kind of basic materials here. So first of all, I want to show you the pills, the little drug pills that you're going to be having. Here's an example of them. Your colors may be different. I have a blue and I have a yellow one. And these are essentially just little bath tablets that kids put in their baths to make the color of the water change. So they're non-toxic, but I will say, obviously, do not eat them. They're not meant to be eaten, but they're not gonna hurt you. So the idea is that you're going to build a drug delivery system so that one of these gets released first, and then after a delay, the second one gets released. Now, what we also have given you is some kind of interesting polymer material here. This is actually a polyvinyl alcohol sheet. It's a very interesting material. You can see it's very firm. It's translucent, and you can see the chemical structure here. So it's a polymer because it's a long chain. But what's really interesting about it is that if you put this in water, it will eventually dissolve. So this is going to be your coating that you're going to use, and you're going to have to make a decision about using this coating. You may also be able to use other coatings. You could use paper towels. You could use plastic bags, but that's not going to dissolve in water. You can use anything else you want to delay dissolution or basically the breakdown of your pill system. But what's interesting is that you could also do multiple layers of this PVA sheet if you want, and figure out how many layers do I need in order to have the delay that I want. The idea of the lab is that you're gonna use two pills at a time, and you're going to design a coating or a packaging of these pills so that the one pill releases in about 5 seconds, and then the next pill releases in about 30 seconds. And just to give you an idea what this could look like, we have a little video of an example of how this could be packaged, how you might want to design it, how you can basically assemble it, and then how you should test it based on what how you measure your design and evaluate your design using your eyes and looking at and timing when these things release. So you've seen an example in the video of how what your task is, and you should think about how could I test how fast some of these materials and these coatings will actually dissolve in water. So what we want you to do, as I said, is to design a system where you have a dual delivery system, where you have a one pill dissolving first, giving the color of water some certain color, and then 30 seconds later or more, have the second pill be released. And then you start to see that color. And then ultimately, over time, both of those colors will be all kind of combined. So we want you to take some good notes, do your experiments with documenting how many layers, what kind of materials you used, and then also your results. And then obviously test your pills and see how it all works. All right. So in this case, if you had a yellow and a red pill, in the end, you should ultimately have an orange solution. And then take some photographs, take some pictures at various times in your process. So here's just some pictures that demonstrates kind of what you might see. In this case, after 15 seconds, after close to a minute, and then finally after 5 minutes, what your trial looks like. This is really just a couple of more formats that you can use for analyzing your results and documenting your results. And again, the goal is to try to figure out, can you get a drug delivery system that releases first at 5 seconds and then finally at 30 seconds. Finally, we've given you some costing. So we want you to think about what is it that you can do to optimize your not only your performance of your pill system, but also the cost. But in the pharmaceutical industry, it's usually quite typical that the cost of the material, the cost of the packaging, the cost of the pharmaceutical drug is very, very small compared to the value and the price that gets paid. And the reason for that is because the pharmaceutical companies invest a lot of money at risk in designing and developing new drugs, and it's a very expensive process. So that is one of the reasons that pharmaceutical drugs are very expensive, because you're not just paying for the drug itself or the packaging, you're also paying for all that development cost, which is what allows us to develop solutions to the medical problems using pharmaceuticals. I hope you enjoy the lab. It's gives you a little idea of how you have to think about packaging, how engineers are involved in that, the design of packaging and the testing of packaging. And if you did like this type of breakout, if you're interested in the pharmaceutical industry or how engineers are involved in the healthcare industry in general, you might want to look more into the topics of drug discovery, customized or personalized medicine, medical devices. As I mentioned before, at GE, my previous career, or one of my earlier jobs in GE, we were making pharmaceuticals, but GE also is a major producer of medical devices for imaging. But so are other companies, Philips and others, Siemens. And then also maybe you'd be interested in genomics. And if you're interested in the areas that types of engineering that are involved in the pharmaceutical or healthcare industry, there actually are a lot of different areas. Obviously, biomedical engineering. I mentioned chemical engineering for manufacturing of pharmaceuticals, and of course, manufacturing engineering. A lot of these medical imaging equipment is material science, and mechanical engineers are involved in that. So there's a lot of opportunities for engineering in the healthcare field. So I wish you luck with your lab breakout, and I hope you're successful in designing a dual release drug delivery system and have some fun doing it. So good luck.
Welcome to your drug delivery system extension. So you've already done the extensive lab activity, and this is just to supplement your learning and provide you another opportunity to learn more about real-world applications of what engineers do relating to drug delivery systems. And so this extension will teach you and provide an example of how to engineer the uptake of medicines in the body. And your goal today is to learn about drug delivery systems, how they work, and why they improve patients' lives. Because at the end of the day, the patient is who you are helping and who engineers help by creating drug delivery systems. Just to introduce myself, my name is Maria, and I attended the University of Notre Dame. I graduated in May of 2022, and my major is chemical engineering, which is very relevant to this lab topic. And so I worked in tandem with Eric, who was also a chemical engineer, to make this lab activity on drug delivery systems. And then subsequently, we worked together to make this drug delivery extension activity. And the motivation of how I thought of the idea for this extension activity is that in my first year of working in the real world as a chemical engineer, I have been working as a process engineer in a manufacturing facility that makes biologic drug substances. So this activity is to give you a taste of what a real manufacturing site looks like, and you're going to put yourself in the shoes of a chemical engineer in the manufacturing site. Today, we're going to go through the extension activity, which will give you a deeper understanding of the topic at hand and allow you to really create your own ideas relevant to drug delivery systems, and specifically in a manufacturing environment. And this is completely different from the main lab activity. It's optional, but it's a great opportunity if you liked the lab activity to dive deeper. Again, the goal is to learn about biologic drug processing, specifically focusing on cellular fermentation. And so overall, biological drug manufacturing is done in a clean facility. And here is a picture that shows an example. You see that the technicians working there are covered head to toe with protective personal gear in order to keep the environment clean. And we need to keep it clean from contaminants like skin cells, hair, germs. Think about if someone sneezes in there, you don't want that getting into all of the biological drug substances or medicine, because at the end of the day, these are going to be consumed by patients. And you can think of it like this, like in any food, you don't want contaminants in it. And just like that, it's really important to keep a sterile environment in processing facilities. So overall, when we look at the basics, biomanufacturing involves engineering a cell to produce a specific protein. And you can code a cell to make a certain type of protein, and the protein will target different biological functions, and you can make a medicine out of this protein. And so we already touched upon, because this is going to a patient, these proteins need to be free of contaminants. And so overall, this picture is a good glimpse of something that you'll see in the real world. It resembles the manufacturing site I work in. And you will also notice that the equipment here is stainless steel. And stainless steel is important for biological drug manufacturing because these pieces of equipment need to be sterilized between each time you make a different product. And stainless steel is one of the only materials that can handle cleaning at very, very high temperatures. So going to the next slide, we are going to dive deeper into the specific steps of biological drug process manufacturing. So the first step is starting out with your cell line. So in the manufacturing site, you would obtain a working cell bank based on the cell line's ability to produce a given protein. And so each drug has a different protein sequence that will ultimately solve the problem for the patient and work inside your body. So scientists will be able to determine which cell line is going to encode for a specific protein. And that's where this all starts. It starts in a tiny, tiny vial and you grow it up from there. So you obtain your cell line in your vial and then you cultivate them and you grow them in controllable conditions. So on the number 2 in the graphic, you'll see that it splits into 3 vials, and then once those grow, you grow more and more cells. They run out of room in that vial, and then you put it in a bigger bag. And so they keep growing in that bag. And then the next step is fermentation. In fermentation, you exponentially grow your cells in number and concentration, and you induce the cells to keep making that protein of interest. So in number 3 in the graphic, you'll see there's a series of what are called bioreactors. And so you put your cell solution in the preliminary smallest bioreactor, which ferments the cells. Your cells keep growing, and eventually they're going to run out of room because they grow so fast, and then they need more space. So that's when technicians in the manufacturing site will switch it over to a bigger vessel, a bigger bioreactor. And now they're happy. They have enough room. They start growing, but eventually they're going to run out of room again. And then the technicians in the manufacturing site will do the same thing over, and they're going to switch it over to an even bigger vessel. And so each process, each drug that's made has specific parameters, and everyone might look different. But at the end of the day, it's just a series of bioreactors growing it up until you have enough to go to the next step. So once you're done growing your cells, the next step would be filtration and purification. And so the purpose of this is to separate your desired product protein from cellular debris, process impurities, and viruses, because there's a lot of garbage in your solution when you grew your cells, because you were giving the cells nutrients. The cells may have split or died. So not every cell makes it, and everything along the way is kept in that bag. So there's things you don't want in that bag and you have to filter out. So that's what the next step does. You separate everything you don't want from what you do want. And what you want is that protein of interest. So this looks like a series of filtration steps. And then finally, after the last filtration step, you only have that protein of interest, which will help form the active pharmaceutical ingredient in your drug delivery system. And so once you have that, you can formulate and package it. Every drug may look different, but this is a step where you make it into what goes into the patient hands, whether that be a tablet or a liquid solution. The final step after you have your protein of interest is to make it consumable by a patient. So this is an overview of the biological drug process that occurs in order to get that medicine into the hands of the patient. So now we're going to look closer at fermentation, and in this video, picture yourself as an engineer in this manufacturing facility, and you'll get a taste of what it looks like to be one.
We all know something about fermentation. It's a process used countless times each day to make a variety of dairy products, baked goods, and beverages. We sometimes think of it as letting foods go bad, but in a controlled way. With a little help, milk becomes yogurt, bread rises, and grains decompose, creating alcoholic beverages and alternative fuels. But looking at these examples only gives us a clue as to what's really happening and how we can use the power of fermentation to cost-effectively create a broad array of biological products. So, what is fermentation? A cell can be thought of as a microfactory. These cells can be bacteria, fungi, or specific cells from mammals, plants, or insects. In biotechnology, these cells are used to manufacture a product in a process called fermentation. For yogurt, buttermilk, and cheese, we use bacteria. To make breads and alcoholic beverages, we use yeast, a fungus. And the production of some vaccines require the growth of mammalian cells that are infected with a specific virus. The product the cells manufacture is usually a chemical the cells contain naturally, or a substance that the cells have been genetically altered to create, or even a metabolic waste product of the organism's growth, like one of our examples, alcohol. There are too many everyday products created by commercial-scale fermentation to even list, but some common ones include amino acids, biopharmaceuticals, dyes, enzymes, food products, lipids, steroids, and vitamins. Fermentation is a reasonably simple process. A cell is selected based on its ability to produce the desired product. A seed stock of cells is put into a small amount of media. Media provides the nutritional products the cell needs to grow. When the population of cells has grown and consumed most of the nutrients, it's moved into a larger vessel with more growth media, and the process repeats. This scaling up is complete when the quantity of cells is large and healthy enough to transfer into a production vessel, often referred to as a bioreactor or fermenter. With plenty of fresh media now available and under tightly controlled conditions, the cells grow and manufacture product. When the fermentation is complete, the product is harvested. Fermentation is known as an upstream biotechnology process. It occurs early in the production flow, before recovery, purification, formulation, filling, and packaging. To better understand the fermentation process, we should first find out a little bit about the cells we use and what they may require to reproduce and stay healthy. Different cells have different needs. Some are aerobic, they need oxygen, while others are anaerobic and do not require oxygen. All cells require nutrition. A properly formulated media contains the necessary nutrients to allow cells to grow and produce. The fermenter mixes the cells evenly throughout the media to suspend the cells and supply the oxygen necessary for growth. Effective and efficient fermentation requires rigorous monitoring and control of the environment within the bioreactor. Key factors include temperature, pressure, pH, which is a measure of how acidic or alkaline the media is, oxygen, usually measured as dissolved oxygen within the media, and nutrient levels. Although the environment and the media are tailored to the needs of specific cells, the life cycle of almost all batches follows a predictable pattern. The growth pattern has 4 phases: lag, exponential or log, stationary, and death. When a cell is first introduced to fresh media, it has to adapt to its new environment. This creates a lull or lag in the growth timeline. After the organism adapts, the batch takes off. The cells begin dividing at a constant rate, an exponential or logarithmic or log increase, doubling, then doubling again, and on and on. As the nutrients in the media are consumed, toxic metabolic waste products build up, cells begin to die, and growth slows. When it reaches a point that just as many cells are dying as are dividing, the batch enters the stationary phase. This is the point at which the key nutrients are completely consumed, the fermentation is stopped, and the fermented broth is harvested. If the fermentation were allowed to continue, the cells would enter the death phase. More cells die than divide, and, similar to the exponential phase, the death rate increases logarithmically. Now that we have a basic understanding of how fermentation works, let's look at an actual process and see how it all comes together. For our sample process, we will look at the production of green fluorescent protein, or GFP. GFP is broadly used as a biological marker. It's a fluorescent dye that's very well tolerated by most cells and doesn't interfere with normal cellular function. In the GFP fermentation process, we'll need to add an antibiotic to protect the purity of the batch, and then, late in the process, a biochemical inducer to turn on the GFP gene. Our materials for this process will include a bacteria seed stock, in this case E. coli, that has been genetically enhanced to produce GFP. The basic ingredients for a compatible media, which include nutrients, stabilizers, an antibiotic, and an anti-foaming agent, and IPTG, which is the biochemical inducer that switches on the GFP gene. The equipment that we'll be using includes a 300-liter bioreactor, a UV-Vis spectrophotometer to monitor the optical density, which is a measure of the concentration of cells in the bioreactor, a glucose analyzer to measure glucose, a key nutrient, an offline pH meter to help track the acid-base balance and adjust online measurements if needed, and a broth tank for our final product. The bioreactor is equipped with a water jacket around the vessel to regulate temperature, and integrated sensors to monitor key environmental factors, including dissolved oxygen, pH, internal temperature, water jacket temperature, and vessel pressure. The reactor also has an agitator, dedicated ports for adding seed stock and media ingredients, separate ports for acid and base supplement, air filters for supply and exhaust, and valves for drawing samples and for harvesting. Most fermentation and monitoring functions can be managed from the bioreactor's dedicated process controller. Before the fermentation process can begin, the area must be prepared. Preparation includes removing equipment and material that won't be used in the process, cleaning and sanitizing the area and equipment, and sterilizing equipment as required by the SOPs, Standard Operating Procedures. Sterilization is used to eliminate unwanted microorganisms which can grow naturally in the fermentation media and process equipment. Also, all required materials and documentation should be gathered and prepared, and all process control software should be loaded and verified. The fermentation batch process will be guided and documented with the BPR, Batch Process Record. The batch record leads the operator through the process, step by step, with each step requiring a sign-off and separate verification. This record also includes spaces for documenting key times, activities, and instrument readings. The GFP fermentation process really begins with the expansion of our bacteria seed stock. After removing the specially modified E. coli from the freezer and thawing it, it's used to inoculate a small amount of fresh media in a shaker flask. After the number of cells has reached the target amount, the thriving cells are ready for fermentation. Meanwhile, in the fermentation area, operators begin with a complete check of all critical equipment. Valves, caps, and lines are checked. Hoses are tightened. Probes are verified and calibrated. And 10 kilograms of HPW, high purity water, is added to the vessel. The bioreactor is brought up to normal process pressure and held there in order to check for leaks. The pressure is monitored over a 30-minute period. If a leak is detected, the problem is corrected and the test is run again. Once the reactor passes the test, we are ready to mix the media in the vessel. The agitator is turned on, and the ingredients are added. Yeast extract, tryptic soy broth, ammonium chloride, sodium biphosphate, monopotassium phosphate, and an anti-foam compound. Once all the initial ingredients are in, another 10 kilograms of high-purity water is added. All ports and valves are closed. All condensate valves are opened. And the bioreactor begins an SIP, Sterilize in Place cycle. The target for sterilization is 121 degrees Celsius for 30 minutes. As soon as the temperature climbs to the targeted temperature, the condensate valves are closed, and the SIP cycle completes automatically. Both the vessel and the media are now sterile, and we're ready to add the final ingredients to our media. The glucose hose is attached to the vessel, the connection is steamed to sterilize it, and the separately sterilized glucose antibiotic solution is pumped into the vessel. Then, a manual pH reading of the media is taken, and the bioreactor is set up for its fermentation cycle. After the inoculation hose is connected to the reactor and steamed for 20 minutes, the expanded seed stock is pumped into the reactor containing the media. Fermentation now begins. The operator takes 0-hour readings and begins to regularly monitor batch temperature, agitator RPMs, dissolved oxygen levels, pH, vessel pressure, optical density, airflow rate, and glucose concentrations. Optical densities and glucose concentrations are of particular interest, so they're graphed as well as documented. When the targeted levels of glucose and optical density are achieved, it's time to add IPTG to the vessel to activate or turn on the expression of the green fluorescent protein in the cells. After allowing enough time for the cells to produce green fluorescent protein, usually 5 hours more, final readings are taken, and a sample is drawn to check the percentage of cell solids. The product is now referred to as broth. The broth, which contains spent media and cells, is complete when the key nutrient, glucose, is mostly consumed and the batch has reached the desired concentration. The batch is then cooled down, pumped into a broth tank, and labeled with the batch number, volume, time, and date. The fermentation process is now complete. The harvested broth will now move downstream to the recovery process, where the cells will be ruptured to free the green fluorescent protein, and the protein will be separated from the other broth components.
So now you got a taste of what it looks like inside a real manufacturing site that makes drugs. And so now we're going to do a deeper dive into the operation modes of bioreactors. So as we saw a few slides ago, bioreactors are a big vessel essentially that your cells grow inside of. So you can just think of it as a container that cells are growing. And cells at the end of the day need nutrients, they need proper airflow, and there's a lot of parameters because cells need the perfect conditions to survive. And so depending on the process, there's different modes of bioreactors. The first mode is a batch bioreactor. So this just comes down to the fact that there's no inflows and no outflows, and you dump everything in at once. So all the nutrients are provided at the beginning of the cultivation with your cell line, and at the end, the product is removed. Another type of mode is fed batch. So in these types of processes, your cells may run out of nutrients after a certain amount of days, and they need to be fed more nutrients. So nutrients are periodically fed into the bioreactor to avoid this nutrient depletion. And then again, your product is removed at the end of the period in which the process calls for fermentation. And the third mode is continuous. So there's a continuous inflow of feed nutrients and a continuous outflow of waste. So the fresh nutrients are continuously fed into the bioreactor simultaneously when cells and waste are removed. So these cells are sent straight to the filtration step, which is done immediately instead of batch and fed batch where it's done at the end. So this does allow for faster processing time. And in the next slide, we're going to go through a few of the pros and cons of each type. Let's think about it. For the batch reactor, where there's no inflows and no outflows, there are some advantages. One is the operational simplicity. It's easy to manage. You just dump everything in, let it sit there, and then take it out at the end. There is not a lot of watch or supervision needed. And there's also a less chance of contamination as nutrients are only added at the beginning. And subsequently after that, the reactor is closed to the environment. And we talked about the importance of keeping your cell line free from contaminants because they are so sensitive. And then another advantage is the separation of batch material for traceability. So from a quality perspective, it's really easy to keep track of everything. And if you have a problem with one batch, you can easily track it down to the one where it came from. Now let's think about the disadvantages. So some disadvantages of the batch mode is it's a short duration, but long downtime to clean the bioreactor in between runs. And the initial conditions are important. They set the tone for everything. So if there's even a slight deviation in the nutrient composition, the batch is compromised. And then finally, there's no removal of cell debris or waste products throughout the batch. So the filtration process will be a little tougher to filter out all that stuff that grows in the full fermentation time. Now let's think about fed batch. This is where you have periodically fed nutrients into the reactor, but you don't take everything out until the end. So some advantages is this could be good for longer processes because cells don't run out of nutrients as fast as the batch. There's less downtime, and it can be used to switch genes on or off by changing the feed. So you can make cells stop growing, or you can even intentionally make them die. And then some disadvantages are it's more complex and prone to contamination since you do have to open up an inlet to the atmosphere. And then just like batch, you don't remove any cell debris or waste until the end, so you're going to have a hefty filtration process. And finally, with the continuous mode, some advantages are you can do really long processes with faster production time than batch because the cells are constantly fed nutrients, and you can also control the growth rate, which is determined by the rate at which you refresh the nutrient medium. And finally, you remove the cell debris as you go, making the filtration process easier. And then some disadvantages are it's just simply more complex. You have things going in and out instead of just letting it sit there. And the nutrient feed use is inefficient and wasted since it is continuously fed. So processes like this likely cost more, and the products cannot be neatly separated into batches for traceability. So it can become a quality nightmare if you don't have a proper and efficient tracking system. But at the end of the day, there's pros and cons to all three of these, and at a manufacturing site, engineers will determine which mode is the best for a given process. So your challenge: pretend that yeast is your cell culture and determine an optimal nutrient formulation for your own bioreactor design. You're going to be a chemical engineer in action, designing a bioreactor and all of your feed inputs and outputs. So breaking it down into steps. Number 1, learn about biological drug processing, specifically focusing on the fermentation step. So feel free to go back to the slides that we just discussed to go through everything a little deeper and reread through things you may have not understood the first time. And then step number 2, design various nutrient formulations to feed your cells. Number 3, build your bioreactors by mixing yeast, warm water, and nutrient into a sealed plastic bag. So your sealed plastic bag is going to be essentially your own bioreactor. It's going to simulate a bioreactor condition, so you can think of this sealed bag as a closed bioreactor. And once you have all of these in place, you can test your formulations and learn which formulation is the best for an ideal bioreactor operation. So let's break it down. Think about the process that we just saw in the manufacturing site. So your process vessel is a bioreactor. Those are the big stainless steel vessels that we saw. Your first input is the living organism, which are cells. It's your cell line specific to the protein of interest. Your second input is nutrients. This is the growth medium that contains cell nutrients so your cells can adequately survive and grow. And then your product is your cell debris and your product protein. In the lab activity today, your process vessel that will simulate a bioreactor is your sealed plastic bag. Your first input is going to be yeast, and then your nutrient input is what you're going to want to figure out is the best way to cultivate yeast. And so you're going to determine your optimal nutrient formulation today, and your product is going to be ethanol and carbon dioxide, because when yeast is given the optimal nutrient, it undergoes fermentation, producing those gases. So let's get to it. It's time to create your own formulations. So here's the procedure. You're going to want to label a resealable snack-sized plastic bag with the formulation number from the formulation table on the left. So this table on the left is kind of like your recipe book. You're going to feed each of these formulations into 4 separate plastic bags. To each bag, add the appropriate amount of yeast and sugar according to the formulation table. So for example, for formulation 1, you're going to label a plastic bag number 1 and add in 1 teaspoon of sugar and 1 teaspoon of yeast. After that, after you have your 4 bags assembled and ready to go with the proper formulations, you're going to add a 1/4 cup of warm water and seal the bag immediately. And one key note is to remove as much air as possible when you're sealing this. So the warm water is going to activate that chemical reaction that produces fermentation. So your reaction is going to start right away. So that's why you want to do this in a timely manner of closing your plastic bag. And then once you close it, just gently mix everything together for about 10 seconds. So your yeast and sugar is adequately spread out. It's not just sitting at the bottom anymore. And once you do that, just lay the bag flat on the table. So do this for each bag. You should have 4 bags on your table, and then you're going to want to monitor these bags. What you're going to do is measure the bag thickness and take observational notes on the provided table at about 2.5-minute increments. And one little warning. If your bag becomes too inflated, please open the bag and release the air to avoid it from popping. So this will help you visualize your setup. So this is just recapping what we talked about in terms of the steps of this lab. So you can see that this student labeled each of their bags with the appropriate number. And then the right pictures show the bags laid on the table after a 1/4 cup of warm water was added and the formulation was mixed for about 10 seconds. So when you're done with the procedural setup, you should have 4 bags laying on the table, ready to go. Specifically, when you're monitoring the bags, you're going to want to take notes at around 2.5-minute increments. And this is a little measuring tip. Place a flat object like a clipboard or another ruler on top of the bag to make it level when taking measurements of bag thickness at each time point. So you can see the student here used a clipboard and then just use that ruler to measure exactly how thick the bag is. And you want to make sure it's level because you don't want one side of the bag to contain everything and the other side be essentially 0 centimeters thick. So just make sure it's level overall when you're taking your measurements. And here is the table where you can record all of your observations. So you see for each formulation, you're going to record the bag thickness at 2.5-minute increments. And just like any engineer does, make sure you take observational notes at each time point. So once you're done with that, you're going to calculate your reaction rate. It's time to calculate the reaction rate for each formulation, and this table will guide you through. So fill in the empty values in the table below, and then you're going to get your reaction rate. So you might be wondering why the experiment inside of your plastic bag produced gases, and it comes down to this chemical reaction. It's the fermentation equation. So when glucose is subject to a catalyst, yeast in this case, it produces ethanol and carbon dioxide. A catalyst is a substance that increases the rate of a chemical reaction without itself undergoing any permanent change. And so when you look at a chemical reaction, you can look at the mole ratios. A mole ratio is the ratio between the number of moles of any two substances in a chemical equation. So if you look here, you just need to balance the atoms on each side. So for example, if you put 4 hydrogen atoms in, you must get 4 hydrogen atoms out. So that's why there's a 2 in front of the H2 on the inputs, and then you count your hydrogen atoms and your outputs in your water molecule, and you have 4 there too. So this just comes down to a balancing act to make sure that for each atom on each side of the equation, they match in number, because you cannot eliminate matter via a chemical reaction. And so the fermentation chemical reaction equation is listed below, and this exercise will walk you through finding the mole ratio for this equation specifically, and then you're also going to figure out how many moles of ethanol would be formed if 1 teaspoon of sugar was completely consumed. So if you need help, ask your teacher, but there are some potentially useful values that will help you in this calculation listed here. And so when you get into designing your bioreactor, your goal is to produce 1,000 liters of gas from your bioreactor operation. Determine what reaction rate would you use and how long does the reaction need to run to meet your goal. These are questions that an engineer would ask themselves when designing a process. And so just think about it. What design formulation led to the fastest reaction rate in your lab activity? How long would this reaction have to run to meet your goal? What is the chemical reaction equation that governs fermentation? So at the end of the day, go back to your table and evaluate which formulation was the best, and this will help you. So this table is going to be your best friend, and it will help you design your bioreactor conditions. So you're given a list of parameters and descriptions, and then you're going to want to fill in your design value or condition. So for example, let's walk through the first one together. For the parameter operation mode, there are 3 modes: batch, fed batch, or continuous. And so batch is where you put everything in, let it sit, and then take it out at the end. Fed batch is where we, in the manufacturing world, periodically feed the cells nutrients throughout the process. And then continuous is just continuous inflows and outflows of nutrients and your product protein. So think about it. In this lab activity, we fed yeast, water, and glucose into a bag, sealed it, and let it sit and observed the chemical reaction going on. So this is a batch condition because we didn't touch it after we fed everything. We just let it sit there and then observed at the end. So I challenge you to go through each parameter in this table and think about what your value will be from your results. And then finally, this slide will help you do some reflection that engineers would do at the end of the process and think about, what did you notice about your optimal nutrient formulation? What type of substance provided the best nutrient formulation to make the most air in a given amount of time? And then how do you think you could improve this? And what are other ways that you could even increase the reaction rate, make your bag inflate even faster? So, for example, one thing to think about is the ideal temperature to grow cells in a bioreactor. And this temperature is about 37 degrees Celsius, but the temperature of lukewarm water was only about 15 degrees Celsius. So that's a little hint of something that you could do next time to make your reaction occur even faster. Have fun with this lab activity and be sure to monitor the process throughout and keep track in your table. So when you are an engineer designing your bioreactor, you can make methodical decisions based off of your results in this lab activity.